Welcome to Introduction to Next Generation Sequencing Analysis, Part 1, Short Read Mapping and Visualization. I'm Phillip Richmond, and yeah, let's have some fun. So a quick workshop outline, brief introduction, preparing your workshop directory, going to go through a short read mapping pipeline and what that means as far as pipeline goes. We're going to learn how to use the tools BWA, which is short for Burroughs-Wheeler Aligner, and SAM tools. Then we're going to analyze an example data set, visualize that data set, and then in the last 20 minutes to half an hour, that'll be more of the open question and answer, where if you have a question, we can you can unmute your mic and we can have a bit of a conversation. Also, in that last half hour, there'll be additional data sets that you can work on, and it'll be a little bit more of a open time to play around. So again, welcome. This is being hosted by UBC's Advanced Research Computing and the WestGrid group, which is a partner of Compute Canada. And as the first session is what is hopefully a useful series, we're really open to things that work, things that don't work, and you know, at the end of this, if you're looking for more info about WestGrid or more info about ARC, go ahead and take a look at these links here. So some learning goals. After this presentation.
Sorry, Philip, I don't mean to interrupt, but I think someone else is sharing their screen right now.
I can't share.
If any of the participants or the TAs, if you can just double check that it isn't you, you'll want to move your cursor, and if you see the icon fourth from the left that is sharing, click on stop sharing.
Mauricio.
Mauricio, can you hear us? Are you able to turn off your sharing?
Okay. Are we back?
I think so. Thank you.
Okay. A little hiccup, no problem. Okay, so just briefly, I guess, on this slide, there are a couple of useful links. No, I'm just going to keep in this space. All right, so some learning goals. At the end of this, I hope that you can learn how to interact with the WestGrid compute environment and queuing system. For some of you, this is going to be new. For others, this is maybe we can teach you something that you don't already know. We're also going to explore some of the command line usage of popular bioinformatics tools that are used really in an abundance of applications. We will learn about file formats, FASTQ, SAM, BAM, and FASTA. Mauricio is back. Okay. I'm just going to kind of continue talking here. Mauricio, what happened?
Just to note, everybody who's watching, you can select which screen sharing you're seeing. So if you go to that 1, 2, 3, 4, 5th toggle, you can choose Philip's sharing, and then you'll be able to see what Philip is sharing. Thanks. So yeah.
Okay, and we're back. So after we learn about some file formats, we will visualize our mapped reads using the Integrative Genomics Viewer, IGV. And at the end of this, hopefully you have gained some confidence in the ability to analyze your own data. So this is going to be an interactive experience. Questions can be posted to the group chat or to the Google Doc. And we have 4 TAs with us today to be answering some of these questions and problems as they arise. Okay, so computing via servers. The user, this is you with your smiley, ready-to-learn face, and you are interacting with your computer. And through that computer, you've opened up a terminal, and once we connect to the Orsinus compute server through a secure shell connection, then you will be essentially on the server, what I've kind of highlighted in this orange box. What you're actually going to be interacting with is the head node when you type things into the command line, and when you want to run jobs or processes that are going to take a little while, you will submit them through a job scheduler, and you will type up your jobs in a job script. More on this a little bit later. So short read sequencing, if you are brand new to this, then I highly recommend going to YouTube and watching a tutorial or a video that explains this technology a little more in detail. But briefly, you have genomic DNA. It is sheared. You ligate adapters, apply to a flow cell, and then on that flow cell, you can have isothermal bridge amplification, so that in little clusters on that flow cell, you should have identical clusters of sequences. And then through Illumina's DD-NTP adding, where you add in the radioactively labeled dinucleotides, they will add one at a time, adding each base pair to your sequencing read. And then at the end, you get your data in the FASTQ format, which we will go over a bit later. Some different applications for short read DNA sequencing and alignment using the BWA and SAMTools pipeline that we're going to be going through. You can do variant or mutation calling. You can look at protein DNA or protein RNA interactions with ChIP-seq or CLIP-seq, 3D chromatin organization, regulatory sequence analysis, or most popular for a lot of people is transcriptional analysis, things like RNA-seq. So let's all get started. This is now going to be where we're getting people to interact. So go ahead and open up your terminal. If you are on a PC, that's MOBA Xterm or PuTTY, and if you're on a Mac or a Linux box, then go ahead and just open the terminal application and log into Orsinus. So I will have this over here. So logging into Orsinus, SSH, your username at orsinus.westgrid.ca. Type in your password, and voila, a message kind of like this will pop up. Okay. So when you first log into Orsinus, you are going to be in your home directory over here. But just a brief view of the Orsinus file system organization all the way back to what is called the root, or this /. So whenever I mention something like full file path, I mean that the file path that you're typing in should start all the way at the root and then be connected to whichever directory or file I'm pointing you at. So while there are lots of different directories at the root, the only ones that we're really going to be paying attention to are the home directory, where we're going to start, and then this global Scratch ARC training directory, where we will be processing and viewing our data. So again, when you first log in, you're in your home directory, and if at any point in time you get lost, you can always type in PWD, or print working directory, and it will tell you where you are. So again, once you log in, you're in your home directory, and that's just a standard place to be. So let's go ahead and explore the workshop directory in Global Scratch ARC Training. A quick note for those of you that are new to Linux, I will often have the commands that I need you to type in order to, for example, explore Global Scratch ARC Training. Most of you have completed the quiz, so this should come as a second nature, but it's nice just to have a little bit of a refresher if you need it. So we will go ahead and change to that directory, cd/global scratch arc training. And if we do an ls in this directory, you can see there are 5 subdirectories: genome, process, quiz, raw data, and scripts. So the first thing I want you to do is go ahead and make yourself a workshop directory inside of process and title it something like your last name. So I will go into process. I will make myself a directory. And by typing ls, I can see the people that have also made directories. Very nice. So once you make your directory, we're going to go ahead and copy some files into that workshop directory. So the files that I want you to take are from global scratch arc training raw data, and these 2 files, NA20845, chromosome 19, subregion R1.fastq, and the, oops, this should be in R2.fastq. Okay, so you can copy both of those with the full file paths using this command here. cp/global scratch arc training raw data NA20845, and if I just hit star and then tab twice, you can see that I've selected the 2 files, and we want to send those to global scratch arc training process Richmond, or your own directory. Usually when I'm copying files, I like to make sure that I have successfully copied them, and it looks like I have. So what are these files? These files come from the 1,000 Genomes project, and they represent paired-end sequencing raw data files. There's lots of data that's available in this format through the short read archive, and if you would like to explore some of your own analysis on publicly available data sets, I highly recommend you go to this website. So the FASTQ, you might also see it as FASTQ or .fq, files will contain the raw sequence reads, as we call them. So these are just the short fragments of DNA that have come off the sequencer. And so for paired-end reads, the files are sorted so that each read in the R1 file will have a corresponding pair in the R2 file. And we can look at the contents of this file using the head command. There are other ways to look at the contents of a file, including more and less, so do whatever you're comfortable with, but I'm going to show you with the head command what this looks like. Okay. So this is the FASTQ file format. So what you're looking at here in the FASTQ file is essentially each read represented by 4 lines. So the first line starts with the @ sign, and then there is going to be essentially the read name. On some FASTQ files, this will be an identifier that lets you know where the read came from the actual flow cell. But in other cases, like for example our file, since we downloaded it through the SRA, this file just has the SRA tag. Next comes the actual sequence. So these reads are 250 base pairs long. So this is 250 base pairs of A's, T's, G's, and C's. Then after that will come a plus sign. This is pretty much just a placeholder. Sometimes it may be plus sign and then the read name again. Most often nowadays it's just the plus sign. And then lastly you have this crazy looking string of what looks like random characters. But in fact, this is an ASCII encoded quality score. So the ASCII encoding system, if you'd like to learn more about it, Wikipedia has a very good article describing this format, which is where I've borrowed these images from. But ASCII encoding is a way to take a single character and represent more multi-character digits. So if we were only to use numbers, we could only have 10 possible quality scores. But with an ASCII encoding, we can have up to, at least in this case, 40 quality scores, which is a standard for FASTQ files coming off the Illumina sequencer. Now, what are these quality scores? Well, quality score corresponds to the probability of error. So obviously, the higher quality score you have of a single base pair, the higher or the lower probability of a error at that position. So if we go ahead and take a look at our first read, you can see the first quality score in this file is the less than sign. So for these reads, the less than sign is a quality score of 27, which is not bad. The quality percent chance of an error is quite low there. But if you look near the end of the read, which is quite common for these much longer reads, especially over 200 base pairs, you see that we start to get a lot of these single apostrophes. And so the apostrophe is a quality score of 6, which is much worse. And those are probably going to be some sequencing errors near the end of the reads. Okay, so the next thing that we're going to need for our mapping is going to be the genome files. So you can go ahead and explore some of these genome files by looking in the global Scratch ARC Training genome directory. So we can go there now. If you do an ls, you can see there are many different files in this directory. The one that I'm going to direct your attention to first is called genome.fa. So I downloaded this from the Illumina iGenomes website, which the link should be in one of these. Yep, there it is. So you can go to this link if you would like to download this yourself into a different directory or onto your own server. So this genome.fa file, this is the HG38 genome. And if we take a look at this file with the more command, genome.fa, you can see that the top of this file starts with the greater than sign and then the name of the chromosome, or contig. In this case, this is chromosome 1, and then some more associated information with that chromosome. For example, showing that this is the reference genome version GRCH38. So if we scroll down, you can see that at the beginning we're just seeing a bunch of N's, but eventually you will get to the actual start of chromosome 1 with the nucleotides. Also within this directory, we have some of these files that are start with genome.fa, but then have different suffixes, including .amb, .ann, .bwt, .pac, so on and so forth. Now, these genome indexes were actually created by BWA using the BWA index command, which I can show you later, but to index a human genome, it takes a little bit longer than we have for this workshop, so I've gone ahead and done it for you. Now, as far as the details as to what is exactly within these files, that's not something that you really need to worry about. If you are interested in the algorithm and interested in how the genome is indexed specifically, then I encourage you to look at the paper describing this method, which is the DOI is linked just at the bottom of the slide there. So, yeah, one thing that we can do is we can copy these genome files into our process workshop directory. So I can do a cp of genome.fa, and if I just type the asterisk, then it's going to grab all of the files that start with genome.fa, and I can copy that to my workshop directory. Now, some of these files are quite large, so this is going to take a little while. So while that's running, I will hop back to the slides. So a bit of pipeline overview. In bioinformatics, when we refer to a processing pipeline, we're really just talking about all of the different tools and software that collectively will take our raw data and give us a final output. So for the pipeline, I've broken it down into 3 sections. The first section is read mapping, where the software we'll use is BWA mem. The next is file format conversion, where we will take the output of BWA mem and create a binary version, and then sort it and index it. And then in the last step of our pipeline, we will visualize this final data in IGV. So the first step to this pipeline is read mapping. So for read mapping, you really just need 2 things. One is going to be your reads, and the other is your genome index. And those 2 are going to combine within the BWA mem tool to produce a SAM file, which we will talk about in a bit. So learning the BWA command, one of the best ways to learn how to use a tool is just to type the tool into the command line, and then that will bring up the tool's usage. So it looks like my copy is still running. I could either open a new window, or I can push it to the background by typing Control-Z BG. And now, from the command line, I'm able to continue working. So again, that was Control-Z, Z, for you Canadians, and BG to push it to the background. So once we've done that, you can load the module. So on all of the WestGrid compute servers, there is the module system in place. So you can look at available modules by typing module avail, and that will print out a list of all of the modules installed on this server. For us, we are going to be using the biotools. So you can do module load module load biotools. Okay, we're just going to log in again. Not sure why. Are you getting that issue? So you should be able to type module load biotools. Module load bio-tools. Okay. Some weird issue that I ran into. No worries. So once you have the biotools module loaded, you should be able to type BWA mem. Now when you type in BWA mem, it will bring up the usage statement, and you can see a long list of available options for when you want to run this tool. So breaking down the initial usage statement, what you're going to see at the very top as usage, it will say BWA mem, and then in square brackets options, and then in these angle brackets it will say IDX base in.1.fq and in2.fq. q.fq. So the square brackets denote optional arguments. This is common for a lot of bioinformatics software. You'll see these square brackets being optional arguments, while the angled brackets are required arguments. So an example command would be bwa mem, your index base, or for us it would just be genome.fa, your reads1 file, your reads2 file, and then by default this will print to the screen, and we can capture that output with the greater than sign and then our file name. So I'm not asking you to type this in right now. We're going to go through the actual commands with our data in just a minute. So the next step to the pipeline, once we have our sample.sam file that's going to come out of the bwa mem tool, we'll be going through the samtools suite, specifically samtools view, samtools sort, and samtools index. So again, these are part of the biotools package. So if we just type samtools, you can see all of the different samtools sub-options, and the first one that we're going to want to try is samtools view. And again, in samtools view, you're going to have lots of different options available, but I have highlighted the options that we're going to use. So samtools view -lowercase b, your input sam file, -o, your output bam file. So what that would look like, samtools view -b, sample1.sam, -o, sample1.bam. Pretty simple. Next is the sort command. So samtools sort will give our in.bam file and out.sorted. And then lastly, samtools index, which will create an index of that file, which is necessary for a lot of downstream applications, including visualization with IGB. So let's briefly chat about the queue. For a lot of you, this is something very familiar, but for others, the queue is something that you may have never experienced, even working on several servers that don't have queues. So the idea of the queue, again, is just a way to manage the jobs that are being submitted from the users on the head node, and then taking those jobs and distributing them to multiple different compute nodes where the job will actually be processed and then the output will be returned to you. So in order to submit a file or a script to the queue, you can type the qsub command followed by the name of the script. If you want to check the status of the queue, you can type show queue or qstat. And so we can go ahead and type show queue. And if you type in show queue, you can see that there are lots of users taking advantage of the WestGrid compute resources. Your username is going to be in this column here. The first column is the job ID, and then the status. So you can see a lot of these are idle. If you keep scrolling up, you can see the jobs that are running by user Granados. And then some details about how long the job has been running and the date that it was submitted at. Now, oftentimes we don't really care about what all these hundreds or even maybe thousands of users are doing, so we can just look at qstat with the -u option of our own username. So qstat is only going to show you things if you have anything running. If I type show q with -u username, then it will give me details on any active jobs that I have, any eligible jobs that I have, and any blocked jobs that I have. So because I haven't submitted anything, this is empty for me. So making your PBS Q script, the best resource for understanding Q scripts, without a doubt, is going to the WestGrid support website and then looking under the running jobs file. And so within this, there are example scripts, and then there are also details on all of the different options that you can have within your Q script. Luckily for you guys, I've already made a script with the BWA mem and samtools commands in it, and so you can now copy this script into your own workshop directory, and we can take a look at it. So I will copy from global scratch arc training scripts map and convert.pbs to global scratch arc training process, and then my own directory. Then if I change into that directory, I can see I now have all of my genome files, I have my 2 FASTQ files, and I have my map and convert script. So we can open this in Emacs or any editor that you feel comfortable with. I will go ahead and use Emacs. In order to use Emacs with the graphical user interface, when you log in, you need to specify -X if you're on a Mac. So now we'll go back to that directory in process. And when I type Emacs and the name of my file, a window will pop up where I can now edit the script. There's apparently a new version of X11 open. Okay, so within this script, the top part is what we're going to focus on first. So the top part of this file starts with a necessary line that's going to tell you this is written in the bash language. Next, whenever you see a # followed by PBS, that is a command that is actually being read by the scheduler. So if you want to make comments or notes to yourself, I recommend using the double #, and then that is not going to be interpreted by the scheduler. But anything with a single # followed by PBS, that is something that the scheduler is going to interpret. So the first thing that it's really going to interpret is that I'm asking for 4 processors. So PBS-L prox equals 4. How much RAM or memory does each processor need? PBS-L PMEM equals 2,000 megabytes or 2 gigs. The maximum wall time in hours, minutes, seconds. And lastly, when the job begins, ends, and deports, I want it to send me an email. And that email is right now just called a placeholder, but if you want to edit it, you can go ahead and within Emacs, we can change this to something that works for you. Please don't use my email, otherwise I will be getting a lot of emails I do not want. And then the next part of this script is where I actually have the commands that we had briefly gone over before, except here I filled them in with the full file paths. So the first command is to make sure that the biotools is within our path. Next, we will map with BWA. So I'm saying BWA mem. I asked for 4 processors. So here, this -T is also asking for 4 processors within my BWA mem command. Then I have the location of the genome and its indexes. And then my read1, my read2, and then I'm capturing the output into a SAM file. So right now, if you were to run this script, it would work, but the output would only go to my directory. So what I want you to do is go through and change these instances of Richmond to whatever is a relevant path for your workshop directory. So I'll go ahead and change each of those. And while I'm doing this, I will just say that I have been using these scripts for many years, and I still will copy a script and use it as a template for a different job, and I will swap out the locations of things like file paths, but that way you don't actually have to retype all of this every time. A couple more things to change. Again, the next part of the script after the bwa mem command, we are going to produce this SAM file, and then using SAMtools view,
Andre, do you have a question to ask? If not, if you could please mute your microphone thank you.
Awesome. No worries there. So SAMtools view, again, -b, our in.sam, our out.bam, then SAMtools sort, and then SAMtools index. And I can save, and then I can close that. And now, if I look at my map and convert file, those changes have all been made. Okay, so now we are going to run our job in the queue. So we can submit this using the qsub command. So qsub and then the name of our file. So qsub map and convert.pbs. And then I can track that job with the -u command. Yes. Show q-u Richmond P. I have one eligible job. It is idle and it is waiting in the queue. So when space opens up for that job to finish, then the job will move from eligible idle jobs into active jobs, which we will just have to wait for that to happen. So while we wait, I can talk a little bit about the output SAM file. Looks like I'm having some kind of an error. We will address that in just a bit. If you look at the my directory here in global scratch arc training process Richmond, you can look at the example SAM file that I've already created. So again, you can go into my directory while we wait, which everyone should have permission to do. And if we take a look at this SAM file, then at the top we have these @SQ lines. Now these lines are describing the individual sequences, so chromosome 1 and its length, and then if you continue to scroll down, the next line after all of the sequences is this PG, and this tells you about the command that you actually ran to generate this. Next is the individual reads. So every line will have its own read, and then these are tab delimited columns. So some of these tabs, the first one is going to be the name of the read. The next is just a flag that you don't really need to worry about. The reference sequence, so where the read mapped. So we're mapping to chromosome 19. And then the position on chromosome 19 that this read is mapped. The mapping quality of the read, which the mapping quality for BWA will tell you about how unique that read is mapping to that location. This is a string that will tell you about the mismatches or insertions and deletions for every read. And then a couple more columns. Again, you don't really need to worry about them for now. Followed by the sequence of the read and the quality score line for that read. And then additional columns with other information. So the BAM file, So the BAM file is the binary format of that SAM file. And so if you were to try and look at that in the same way as you look at your SAM files, then I only recommend only doing this once. What you're going to get are essentially characters that are pretty much randomly placed there because you cannot look at these files in the same manner. So if you want to look at the contents of a BAM file, you need to convert it back into SAM format. Okay, so within our script, we had both read mapping and file format conversion, and I will debug what was happening with my script in just a minute. For those of you that were able to get it to run, you can copy your own files down to your own laptop. And for those of you that were unable to get it to run, including myself, I have these backup files located here for you to copy down. So the file transfer client that I have recommended is called FileZilla. So if you open up FileZilla or whatever your file transfer client is that you'd like, we can copy some files down to our desktop. So in the top here for host, this will be orsinus.westgrid.ca with our username. Enter your password and just type port 22. This will connect us to the server. And now on the right side of my screen, you can see that I have connected to this location, which is my home directory. And if I just type here in the remote site, I want this to be global, scratch, arc, training, process, and Richmond. So if I type that, now you can see that these are the files that are listed in this directory here. So the files that I'm going to want to copy over are the NA20845 sorted BAM and the sorted BAM.bai. So this is our sorted BAM file and the index. And I can just click these and drag them over anywhere on my computer. I will go ahead and put them into the We'll just put it into desktop. And now those are successfully transferred. So now if you've done that, the next step is going to be to open up IGV. Any questions in the chat? Okay, cool. Yeah, any questions that are just burning, go ahead and ask them in the chat. I must have edited my file in some wrong manner, so I was unable to get my jobs to run, but hopefully some of you have gotten it to work. If not, we can debug that. So when you open up IGV, you can select the genome that we're going to be using. So for us, it's going to be the human HG38. If this is not one of your options, you can find it by clicking on the More button. But for me, I already have it loaded, so it will just be there. And I'm going to want to load those files that I had just brought down onto my computer. So if I go File, Load from File, it's got to think a little bit. And then on my desktop, I have these two files. The only one that I'm going to want to click on is the sorted BAM file. So I'll click on that. And this is a very small file, so it'll quickly be loaded into IGV. So right now, I'm going to get this down. You can see, if you're new to IGV, across the top we have the different chromosomes. You can type a region or a gene into the text box here, and that will take you to that region or to that gene. This track here at the bottom represents the different genes, and when we zoom in, that will make more sense. And so I believe I have directed you to a given region. So again, if you're lost or if I'm moving too quickly, just File, Load from File, select just the sorted BAM. And in the search box, we are going to type this location on chromosome 19. And when I do that, I am now zoomed in to this region here on the left arm of chromosome 19. And you can see that the track, which was white, now that we've zoomed in close enough, is displaying the actual reads for this region. Down here at the bottom, we have the individual genes. So this is the STK11 gene, ATP5D, and if we want to zoom in on any of these, we can simply use this region here to click and drag. So now we've zoomed in. We can zoom in a little farther. And as you continue to zoom in, you can see the individual reads. So these are the 250 base pair reads, and one of the things that is quite apparent when you are zooming in this close is that some of these reads have lots of these individual mismatches. However, a lot of them, which are shaded quite lightly, have been ignored due to the fact that the sequence quality is so low for those individual bases. So again, if we zoom back out, there's lots of things that we can look at. Up at the top, we have our coverage track, and if you zoom in on any one of these locations in the coverage track where there is a colored setting instead of just the typical gray, it will tell you the distribution of A's, T's, G's, and C's over that position. So at this position here, it looks like we have 17 C's and 11 G's. So this is probably a single nucleotide variant in this individual. Some different coloring options. The red and blue just shows the different strands. You can color alignments a variety of ways. You can use no color if that's what you'd like, or you can color them by the read group or the sample. If you're viewing multiple different samples at the same time. Again, zooming in and zooming out, very simple. And yeah, the region that I have sub-selected for us is quite short, so if you go beyond the bounds of this region, you can see there are no reads, and that's just so that these files are manageable. If we were dealing with whole human genome BAM files, then this workshop would take several weeks. So other than that, that is the end of our course right here at 11 o'clock. In the remaining time, I have other raw data files that you can process and analyze. They just represent different individuals that have been sequenced from the, as a part of the 1,000 Genomes project. You can also try to use different options with BWA-MEM. Some things that you may want to try are suggested here, and you can look up what exactly these mean in the BWA manual or by typing BWA-MEM into the command line. And you can also visualize multiple samples at the same time in IGB once you have gotten them mapped. Thank you for all participating, and we will contact you in a few days with a form that will have some room for feedback, what worked, what didn't. This was the first time we've ever taught a session like this, so we're open to any comments or criticisms. The technical issues are just something that I guess happens when you're dealing with this video conferencing style workshop, but I think everything went pretty well. And if you're interested in getting information about how to get a full account on WestGrid for future analysis, that will also be in that email. We hope to see you all at the next workshop, and perhaps in that feedback form, any suggestions for topics to cover, including things like variant calling, which we would be able to identify all of these individual mismatches and which are truly variants and which are just sequencing errors, or things like RNA-seq and ChIP-seq. Those are also things that we can teach within this workshop series. Hope to see you all at the next workshop.